Yes, so hello everyone, and thanks for the introduction. So I will talk about microbiome analysis tools in Chipster. And I think from Aya's presentation earlier, you already got something of a look at the Chipster interface. So my slides don't really focus on that, but they focus instead on this new tool set that we've been developing. But just to recap a little bit on Chipster anyways, so this is a platform for open source data analysis and service that spans a lot of different use cases and workflows. Something that we've been lacking for a long time though is tools for analyzing structure and composition of microbial communities. So um, and this is all based on sequencing data. So this is a kind of gap in the current toolset that we targeted, and at this point, the focus is on amplicon sequencing data, so data generated using DNA metabarcoding approaches. Just a little bit of background on this, though. So the kind of motivation behind this was to make it easier to bridge this gap between wet lab research and then dry lab or computational research. And this can be quite challenging, I guess, for many wet lab scientists, because the computational side of analyzing microbiome datasets often requires a lot of well, bioinformatics skills and then also programming skills, so things like command line usage and so on. So there's a little bit of a knowledge gap. But just to go through the kind of metabarcoding workflow in some detail is we would first collect some data from an environmental sample, extract DNA from there, and then you would use PCR-based approaches to target some specific marker gene. So for example, the 16S ribosomal RNA gene. So that's used as a kind of universal marker gene for identifying bacterial species, for example. And you're then able to run your high throughput sequencing analysis after that. And at that point, you have this usually quite large sequencing datasets that you then push through some bioinformatics pipeline. So the tools and chips to target this bioinformatics pipeline side of things and species identification. And we also have some options there for statistics. So we do the data pre-processing using Mother software, which is well-established software for working with these types of data. That was already in Chipster, but then we've integrated data produced with the fileseek R package and also a number of other R packages that make it possible then to do some further sample processing. So we have options for data tidying, transformation, visualization, and then also statistics. So things like global tests using distance matrices, and then also differential abundance comparisons using this DSeq2 tool that was originally developed for the RNA-seq community. Just to go through the workflow very briefly, we have input files using the Mother software, and then it's possible to import the data. So Chipster also has a feature called phenodata, which is something you can use to specify sample groupings and so on. So kind of like metadata for whatever experiment you're running. You can use the new tools to calculate diversity indices and to perform different types of data overviews. So there's a lot of different tools for that. You can also verify your samples if you want to. That's optional. There are many tools for data tidying, so filtering out noise from the datasets, for example. Data transformation, so there's different options for that. Relative abundance. One is also CLR, so central log facial transformation. So this is a compositional data transformation. So there's increasing evidence that these are useful for microbiome data analysis in particular. We can also convert data to the DSeq2 format. We have options for creating plots or relative abundance plots of different taxa. And then we have these a distance matrix construction options. So we have different ways to calculate taxonomic distances. So for example, Brake-Hurst distances, Aitchison distances. We also have non-metric multidimensional scaling ordinations, canonical analysis of principal coordinates, so these types of ordination options. And if we want to compare communities, then we have the significance tests like PermNova, PermDisp, and then post hoc tests, so Tukey's honesty significant difference test. On top of that, there's the DSeq2 test for comparing taxon abundances. And these are just a couple of different plots that you get with the different tools. So left we have an ordination, so this is a non-metric multidimensional scaling ordination. You can also see the sample IDs associated with the different data points. The image I have on the right is some output from the DSeq2 tool, so that lets you visualize which taxa in particular are differentially abundant between different samples. So not going into that in much detail, but just wanted to show how these things look. So for feature directions, we're looking at adding some analytical functionality. So this is the first iteration at the moment. So something, for example, we want to bring in is paired sample designs. So that would be quite good for us, I think. And then collaborating with different research groups to incorporate new tools. So I think Leo Lahti is here. We hope to work with Leo in the future, for example. And also something we want to explore is different tools for larger scale microbiome analysis. So at the moment, all the tools we have are CPU based, but we could potentially have some GPU tools in there as well, and different machine learning methods. So there's plenty of scope, I think, for future development work in there. So those were my slides. If anyone has any questions, then I'd be happy to take those. Thank you.